Okay, I think we'll do a little video today on the reflex arc, as it's a nice summary of a lot of the things we've already talked about. So let me start with sketching out the basics. Now this is a reflex arc that would be a spinal reflex arc. There could also be a cerebral reflex arc, a brain centered reflex arc, information coming into and out of the brain as opposed to into and out of the spinal cord. I'm going to use the spinal cord as an example. So let's start with a little bit of a cross section of the spinal cord, and we've got our gray matter. So I can get this reasonably symmetrical, right? The gray matter surrounded by white matter tracks, right? The central canal there in the middle, and this would be the posterior aspect, dorsal aspect. And so what I want to do is I want to talk about again, we've been talking a lot about individual neurons and what individual neurons do. And yes, we've seen synapses between neurons, but I want to put it into context so that we can talk about sort of the conveyance of information through the afferent portion of the PNS, and then out of the CNS through the efferent, right? E for exit, to generate motor function, right? And so let me color code our cells here a little bit. Hopefully you can see this well enough. This is a different setup, obviously, now that we're distance learning here, and I'm not doing this from my office. But what we would have, so spinal cord section, and again, this is sort of like a, right, if we cut through the spinal cord and then turned it and looked at the end of it, right? And so what we have is, I'll sort of exaggerate the size of this so we can see things, right? We'd have a sensory neuron somewhere around the periphery. This is what we call the free nerve endings of that sensory neuron, but literally they're just the dendrites, specialized dendrites essentially. Then we would have the axon. We would have the cell body that sits along that axon, and it would come into the dorsal gray horn here, and we would have a synapse. And again, I'm trying to make this big enough so you can see it. Right, that's my synapse. Put a nucleus in our soma, right? So this was our unipolar neuron sort of anatomy, right? The soma has things only coming out of one end of the cell, right? Uni for single. As opposed to the multipolar neurons that we keep drawing over and over again, which we'll get to in a second, because that's the structure of motor neurons. And so let's complete the arc here, if you will, and then we'll start to talk about how, this arc sort of relays information and processes information. In the middle here, between, and usually, but not always, there is an interneuron between, so cell body, this short little interneuron. I wish I could zoom for you a little bit. All right, but I'm drawing an interneuron there between the sensory neuron coming in, delivering information inward. All right, so let's put an arrow indicating the information. I'll put some more labels on this in a minute. And then the motor neuron, which it's going to synapse with, in either the lateral or ventral horn, depending upon what type of motor information it is. Basically the big difference being, is it somatic motor or is it visceral motor? And so we're not really concerned about which it is right now. So we're just going to leave it generic. But I've got my soma, and there would be dendrites on there, just I'm drawing it so small, we're not indicating them. And then we would have our axon exit to come out and synapse. And again, it would probably have branches. Again, remember, this is my shorthand version of a neuron, right, where I sort of draw it like this. I'll put one down here at the bottom, where it's soma, axon, and then synapse, right? And if you like, we can add some neurotransmitter being released there, right? I don't usually draw the dendrites on it just for simplification. But again, this is a completely different neuron structure than the motor neuron here. And so information in this guy is coming out, right? So afferent in, sensory, interneuron, processing. And again, it wouldn't be one sensory neuron synapsing on this interneuron. I'm simplifying here, right? So this guy would do the integration, sort of add up the input to decide, what we need to do, and determine whether or not we need output to this motor neuron based upon the magnitude of the input here. And again, whether it's stimulatory, it would be EPSP, or inhibitory, IPSP, input to this interneuron. Well, if the EPSP input wins and is dominant, we could reach threshold and stimulate this guy, right, and generate output to, and I'll put, let's see, how do I want to do this? What I'll do is I'll do a muscle in cross section. How about that? So if this is a muscle, innervating a muscle, right, that being the whole muscle there in cross section, the muscle is formed of cells running lengthwise, right? So this is a cross section through the muscle. We've cut it. We're looking at the end of the muscle. Well, the muscle is made up of lots, and again, there would be others that are not necessarily innervated by this neuron, right? But lots of skeletal muscle fibers, and again, on and on and on. But you see what I'm getting at here is that we've got this whole muscle with muscle cells that are running lengthwise along that muscle, and then this neuron is coming in and innervating some subset of those muscle fibers, right? So the little red circles are muscle cells, which are actually cylindrical, right? Kind of like the muscle would be longer. Again, we're showing you in cross section. Cut the muscle in half and look at the end of it. Okay, so let's add some labels here, right? And again, let's label this as our reflex arc. And again, why are we doing this? Again, so we don't lose context of how neurons are talking to other neurons as opposed to just what single neurons are doing, right? And so we've got the PNS out here. This is CNS. Again, some of our reference points here. This would be the afferent division, which is sensory. So this is and this would be the efferent, which is motor, right? And again, afferent and efferent divisions of the PNS, okay? Okay, so let's start with our reflex arc here and see if we can walk through the mechanics of what happens at each individual segment in each individual neuron. And so let's put this as a stretch receptor, a stretch receptor, a receptor that's in the muscle, right? So we could have our muscle, let's do this, have our muscle, we can sort of see it going off into the distance here, right? Right? And this guy is innervating this muscle. Let me extend these a little bit. They look like they're more in contact with our muscle, right? And again, these are essentially the dendrites of this sensory neuron. They're called free nerve endings. And they can be associated with other structures. There's what are called encapsulated nerve endings, but we don't need to get specific about that yet. But these guys are where we would either initiate or not initiate the generation of an action potential in this axon. An action potential and then a nerve impulse, right? A series of action potentials traveling along that axon. Now, I want to label one section here. I'll highlight it by making it dark, right? And we could call that the axon initial segment, right? Because we've got this region where we transition from graded potentials, the generation of graded potentials, to the generation of an action potential. And that happens at the axon initial segment, which integrates the graded potentials. Now in this scenario, the graded potentials would be generated by, I was telling you about stretch of the muscle sort of being, we can determine, right? We know where our limbs are in space because our body tells us, gives us information about our joint position as well as the tension and length of our muscles. So having this as sort of a length detector of our muscles. And so what it's going to be is it's going to have mechanoreceptors, right? Remember these different versions of gated receptors, gated channels? These would be mechanically gated channels. In other words, stretch of this muscle would distort. These guys would actually be sort of embedded in the muscle. I'm not showing that, but they would be sort of embedded in the muscle, and stretch of those of that muscle would distort those nerve endings and open mechanically gated channels. Well, if those are mechanically gated sodium channels, we're going to generate EPSPs in these dendrites, free nerve endings. If we generate a big enough EPSP, so let's just write that. So stretch occurs, right? EPSPs would occur because of it. Again, because of mechanically gated channels activated by that stretch. Graded potentials, remember what would conduct, I shouldn't say conduct, convey that's the EPSP to the axon initial segment? Electrotonic current. Is it decaying with distance the farther we go along these dendrites? Yes, it is. Right? Electrotonic current decays with distance. This is where the length constant comes in, right? The farther this electrotonic current travels will be dependent upon the size and resistance of the dendrites themselves. In other words, big fat dendrites would actually convey electrotonic current better than little skinny ones. And again, because it's distance determined, right? EPSPs generated closer to the axon initial segment would have a greater effect because they wouldn't dissipate as much, decay as much over a shorter distance. Okay, well, now we stretched it. We got our EPSPs. Let's assume that we reached the threshold of voltage-gated channels here, which means we're going to get an action potential, right? And our action potential will be regenerative because it involves the opening of voltage-gated channels. And opening voltage-gated channels causes the opening of voltage-gated channels, which causes the opening of voltage-gated channels. It's a positive feedback loop, right? And so we're going to get an action potential. We open voltage-gated sodium channels. We get electrotonic current. We get another action potential. We get electrotonic current. We get another action potential, right? And again, this sequence of repeated action potentials regenerated along the axon is called a nerve impulse. And so we've got this nerve impulse that's now traveling along this axon at high speed. Now, what will cause it to be faster or slower will be the magnitude of Rm and Ri. Now, what I've drawn here is an unmyelinated axon, and some sensory neurons are unmyelinated. That would be continuous propagation. In other words, the action potential would be regenerated at every segment of the axon, right? If I had myelin on here, we would have saltatory propagation. Myelin increases Rm, right? Let's put our length constant up here. Length constant is proportional to, approximately equal to, Rm over Ri, right? And so if Rm goes up, our length constant gets bigger, and our electrotonic current goes farther and faster. And so, again, if I add myelin, this would be faster. And if I made it bigger, which would reduce Ri, right, made it bigger in diameter, that would reduce the internal resistance. And again, reducing this makes this bigger, and again, better electrotonic current. So we've got a nerve impulse that's traveling, and it's going to blaze right past our soma. Soma doesn't get in the way of any of this. And it's going to come into the dorsal gray horn. Actually, and by the way, you may have seen it in lab. These cell bodies all clustered right outside the spinal cord form the dorsal root ganglion, right? And a ganglion is just a clump of cell bodies in the peripheral nervous system. And so we have this clump of cell bodies outside the CNS, all sensory, that form the dorsal root ganglion. Note, we don't have a cell body down here, so you don't have a ventral root ganglion, right? The motor neuron cell body is actually found inside the CNS, not outside. Okay, so we get to the interneuron, right? So, and again, you're not going to be able to see it, but I'm going to have some neurotransmitter release here. What's required for that neurotransmitter release is depolarization of the synaptic membrane. Remember the synaptic bulbs or synaptic terminals? That membrane has to depolarize. We will then open voltage-gated, a new type of voltage-gated channel, voltage-gated calcium channels. Talked about this in the synapse video. The calcium influx would result in the vesicles, the neurotransmitter-containing vesicles, to fuse with the membrane here, fuse with the synaptic membrane, right, of this red cell, the sensory cell, and neurotransmitter would be released into the synaptic cleft, diffusion, bind to receptors on the postsynaptic interneuron, generate, again, could be an IPSP, but in this scenario, it's probably going to be an EPSP. And now our interneuron has the potential to generate an action potential. And of course, then we're going to get action potentials traveling along this very short axon. And by the way, that axon is very likely to split, right? And it tends to what's called decussate, crosses over. And it's going to cross over, and then it's going to run north through the white matter tracts of the spinal cord. And you're going to become consciously aware of, or your brain is at least going to be, get the information that this muscle has been stretched, right? Because that was our initial stimulus. Okay, well, let's follow the action potential. Again, so the action potential is going to just go both directions, right? Conveying information both to the brain and to this motor neuron. We can just repeat the events here. At the interneuron synapse, we get voltage-gated calcium channels opening, synaptic bulb fusion and neurotransmitter release, diffusion across the synaptic cleft, and EPSPs generated on the motor neuron. And again, there could be other cells that innervate this motor neuron that generate IPSPs. Could you consciously avoid contracting this muscle when it's stretched? Yes, if you were thinking about it, that would involve other input to this motor neuron, not the simple reflex arc we're showing you here. Right? But, okay, leaving that out of the picture, our soma and dendrites here would receive information in terms of the chemical signal, the neurotransmitter from the interneuron. New EPSPs, right? New electrotonic current or electrotonic current in these soma and dendrites. Where's the axon initial segment? Right at the base of the axon there, right? Because remember, this blue guy is a multipolar neuron, right? That's the one where it looks more like this, right? Where here, I'll add the dendrites on it, right? And the axon initial segment is right there, right at the very beginning of the axon. So now we have action potential generation in this guy, right? Because we reached the threshold, again, we can potentially reach the threshold of voltage-gated channels here. Sodium channels first, then potassium channels, right? Depolarization, followed by repolarization. Action potential after action potential, right? And off we go to our target. We're going to release neurotransmitter for the same reasons we've talked about before. And now we're going to be stimulating, in this scenario, stimulating these muscle cells to contract. So really, the reflex arc is a lot of the same things sort of over and over again, right? Graded potential, action potential, synaptic release. Graded potential, action potential, synaptic release. Graded potentials, action potential, synaptic release. Over and over again, right? So this is neurons talking to neurons talking to neurons. Now, this is the most simplistic version of a neural network, right? Neuron, single neuron to single neuron to single neuron, right? We use it as an example just to sort of convey how the neurons are talking one to the next to the next. Keep in mind that can be competing information, right? EPSPs and IPSPs coming in together to sort of, determine control of motor neurons. And you go to the CNS, and these become much more complex. with potentially thousands of neurons involved in integration, sort of determining what happens. And again, we can do more complex things and generate motor output, right? Memory and learning and things like that are far more complex than just generating motor outputs. But stimulus, integration, and response does explain a whole lot of physiology. Explains movement of your skeletal muscles, movement of you. It explains control of your, partially, control of your blood pressure, control of contraction of smooth muscle in your GI tract and respiratory tract and things like that. So it does play a major role. This kind of simplistic reflex arc plays a major role in neural function. Okay, I think we did a nice little summary there. What I'm going to come back and do is another video on neurotoxins so that we can see how disrupting functions at a given location play a big role in determining how neural networks work or don't work. It's just a good example of a good way to determine make sure we understand the importance of particular components involved in gradient potentials, action potentials, and synaptic release. Okay.